First of all, thank you very much for having me. I really enjoyed company of like-minded people, as I said. I think of this talk as introducing myself and giving people a reason to find me and talk to me, or avoid me and not talk to me later. I'll give you reasons for both. So I'm Leon Peshkin. I work at Harvard Medical School Department of Systems Biology. Systems is important. It's gonna affect many things that I do. And this year I'm actually visiting Paris Institute Curie for a while. So this is an arbitrary picture of timeline of various anti-aging products. It's not important. I just want some placeholder slide to say I decided to go against the trend and not to write a book about aging and longevity. Because I think nobody would want to read this book. I focus on the negative, maybe excessively.
You're a passionate pessimist.
I am, thank you very much. The term has been coined. I'm a passionate pessimist. I'm passionate about longevity and rejuvenation, and unfortunately pessimistic about many things which are happening in this field. So I just want to think together with you a little bit of something very important and I think disabling to our field. I think that if I were to start a company whose goal would be to show that senescent cells as a category make no sense, or Yamanaka factors actually don't rejuvenate anything, or methylation clocks don't at all measure what we say they measure, probably this company would not fly. If I were trying to publish something like this in the scientific literature, the papers would be read by very people who review my other papers and review my grants. So I have no incentives, not in science, not in the industry, to focus on the skepticism. Yet, I am in this field, I talk to people, and I know that there are very serious objections to what is sort of in the mainstream and what gives rise to hundreds of startups. So I believe this is a problem, but possibly we could sort of get promising directions for rejuvenation from the dumpster of longevity field. And I'll explain what I mean with one example. So, of course, this is something I published on lifespan.io, which I call controlling the controls. So this is general discussion of how we keep publishing results with the lifespan curve of various model organisms. And we would celebrate and consider many drugs, as we did in this session, which extend the average lifespan by 15%, yet it is very well known in the community, but not published, that 15% is just a margin of error. That between large controls, between different locations in the intervention testing program, there is just variability of 15% between controls. Right, so here we're doing a little bit of our own work, where, just as example, we take a popular strain of mice, which is used in many longevity studies, and perturbation is not important. It could be gene knockout, overexpression, drug, diet, it doesn't matter. We ignore most of the paper, we just go and we take controls. So here are all the controls, same mice, same gender, same diet, same everything according to the methods. Look at that variability. This is just half of the slide, right? So what we do then is we superimpose results for several sort of visible studies done in the same mice, right? So if you take Barbarian, you see that both the control and condition are way below normal for this mice. Now, here's probably the most important part of my talk. Do I suggest that we now cross this out and forget? No, this is golden data. instead of this infinite loop of trying and leaving and forgetting a candidate, we should actually cherish this data, because as systems biologist, I want to get information from all of this put together. Right, so here is sirtuin-6, another recent kind of sensational result. Again, control, condition, so maybe rapamycin holds water. Maybe it's interesting. Again, the effect here looks very small. None of this is interesting to me. What's interesting is that today, in the modern time, we do not have a standard for lifespan format where the data would be mandatory submitted in a certain format, where you could easily assemble it in one place, where, you know, if I publish tomorrow paper saying I had this one mouse live 5 years, that would be very impressive with just one mouse, but that's enough, right? But if there is a paper with this sort of result, I want to say, okay, how long do these guys normally live? Show me some right, easily, and that's impossible. So this is one project where we're trying to formalize lifespan data format and suggest a way to assemble and reanalyze this data. This is completely different project, but in the same spirit. This piece came out a few months ago, describing that I'm trying to create and release into the world the platform for, as I learned here, called DeSci, Distributed Science, crowdsourced science. So that the whole cycle of collecting the data of perturbation and publishing it is shortened to days from many, many years, so that many more people can do this experiment and can collect data, and it would instantly go to the central service and be analyzed. So we're having some discussion with distributed science people about that. Right, so again, if you want to get depressed about any of your favorite model organisms, find me later. I claim that for trying perturbations, lifespan longevity interventions, long term, not in an hour, not instant effect, none of the traditional model organisms work for many, many reasons. The data can pretty much be ignored about life extension with intervention in any of those. So I was involved in trying to introduce new species that I'm enthusiastic about, which maybe, okay, the movie is not going to play, that's fine. These are small freshwater crustaceans, Daphnia, which check many boxes. They're very transparent. The innate immune system was discovered in the species. There is a beating to camera heart. They're super sensitive to small quantities of drugs. They're clonal. The genome is available. All of the homologous genes for longevity groups, families of genes are available, and so on and so forth. So, let's see what happens. I lost control over this.
Just go ahead. Let's figure this out.
Right, so this is very much in the same spirit. I do not want to look for the elixir of life. That's not at all the point. I want not to look for new drugs. I want to take old drugs, boring drugs, where we have characterized molecular targets, and compare all of these drugs, some of which are slight poisons and some of which are slightly beneficial, using the same very repeatable baseline to collect a large set of data. Then this data would be amendable for reanalysis, maybe for machine learning methods, for inference and reverse engineering of which modules and pathways are important. So we developed and published this platform and applied it. Here is a very recent development. Somebody heard my talk, Rebecca Sandlin from MGH, and came to me suggesting that we collaborate in applications for cryotoxicity, right? So cryotoxicity is when you want to freeze organs or organisms, and you try to cryoprotect, cryoprotectants themselves are toxic, possibly. So you want to have platform like the one we're developing, where you don't just look at whether this organism is alive, but at many endpoints, reproductive, metabolic, cognitive, if you want, how those are affected in many combinations of cryo preservation and protection against toxicity. So they principally worked in mosquitoes, but apparently in organs and mosquitoes it's just prohibitively difficult, expensive, not scalable, and Daphnia checks all of those boxes. There is another movie here where they actually did the magic. Daphnia, never before in millions of years, was frozen and then awakened alive, and they managed to get initial protocol going where this freshwater crustacean wakes up somewhat alive. The heart is beating, it moves for a little while. So it's very exciting because now you can make next steps. Right, so now on to rejuvenation chapter. This idea that somehow embryo contains the essence of usefulness is a very old idea. My grandmother mentioned to me that she applied face lotion with extract from chicken embryos. Right, so this it's a very intuitive idea, but I don't think we know how this magic happens, how you get young organism from an old egg cell. And I don't believe that Yamanaka tells us how it happens, because essentially it's not about the DNA at all. When you move the old DNA, you move it into an oocyte. So it's not the genome, it's the rest of the cell which does the magic. And I would like to understand this magic better. So this is Cynthia Kenyon's seminal work, where she looks into a worm and shows by imaging methods that there's junk in oocyte, but in the matter of maturation, if you want, this junk is removed. And she suggests that it is removed by some regime of lysosomal activation in worms. Now, there is a short chunk in that paper, little notice about xenopus. They wanted to see what happens in xenopus. Why? Well, first of all, xenopus naturally is much closer to humans, but then you don't just want to image, you want to understand better the molecular mechanisms of what's going on, and they noticed that lysosomes are actually activated in frog oocytes as well. So what are the questions? Very basic questions about this maybe miracle, right? And if there is no miracle, I would also feel we accomplished something. We crossed out definitively direction and learned from it, right? But the questions are there aggregates in vertebrate oocytes? Are those aggregates even a problem in general? Which proteins aggregate, and is this actually cleansed? And if it is, well, how can we somehow apply same mechanisms in somatic cells of adult? Right? That's not how evolution meant it, but we could take this module and hijack it and replace, just the way we took modules for painkillers, antibiotic, immune system, and vaccination. We sort of observe the miracle in nature and move it into a new context, right? That's the idea. So why I think that we are specially positioned to work on this. There's a funny aspect. No one in the world has a large collection of old frogs. We're connected to National Frog Resource, which have been producing transgenic frogs for 10 years now, and they have a bunch of frogs with birthday, including very old frogs, which are important here. there is people, I think, are generally and very unfortunately skeptical about going back and forth between model organisms and human, right? But it's unfortunately just ignorance. Yes, many things don't go back and forth, but many things do. Many molecular mechanisms, many drugs are movable. And so I think this is underappreciated how close some of this reproductive biology is. We have just put out a preprint and will publish soon this work in Xenopus with Steve Horvath and Vadim Gladyshev, where we look at DNA methylation clocks and aging ground zero as happening in young embryo in Xenopus, again, because Xenopus is very specially positioned for such science. This is actually my old work from many years ago. The only point here is that if you look at swimming tadpole with many differentiated cells and you ask about a protein molecule, you reach in and you take a protein molecule out. Was this protein synthesized during development from the oocyte, or was it deposited? Shockingly, it seems like most molecules in this functional organism are not synthesized, they're deposited in the oocyte. So if you deposit junk, you cannot get functional organism. This is not a proof, but evidence that yes, you need to do some housekeeping before you get fertilized. This is just eye candy. This is what frog ovary looks like, and some of the early stage oocyte where you can see Balbiani body, so-called germplasm. Those are large objects. You can manipulate those, you can micro-inject those, you can do things impossible in most other organisms. So they're very nice gradual stages of oogenesis that we've been working through. And just a very general idea of how we look at aggregates. So this is very current work in the lab. The idea is that you collect oocytes and run through a size exclusion chromatography column. You collect fractions of proteins, which should come out in a certain order. But then you identify that some of the proteins are misplaced. They're not in the right context. So why would small molecular weight proteins come out together with a large one? Because they are aggregated. So it's a very simple protocol. At the end of this, everything goes to quantitative mass spectrometry. I don't know why nobody developed this, but of course the devil is in detail. And there's no reason to just look at ovary and oocyte. In principle, it is interesting to profile aggregates in many tissues, and I mentioned we have collection of old frogs. Again, this is something we're developing, a careful guide of blood perfusion in the frog and dissection. I mean, you would be shocked how much frog is a little human, with some exceptions, in terms of structure of the tissue. So we want to look at aggregates in all organs, but why this fixation on oocytes? Well, proverbial miracle of cleansing only is happening in oocytes, if it does. I think that generally frogs are very underappreciated as a model organism. It is much closer evolutionary and morphologically to human than zebrafish, and so generally allows to go back and forth much more. So this program, I think, provides a very nice rejuvenation roadmap, focusing on this specific kind of cleansing hypothesis. We will answer questions about possible mechanism of rejuvenation in germline. We would establish ways to assign age. If you want, you can call it clock, in a different way, looking at proteome in the oocytes, get insights into the mechanism, and it does provide us with a way to look for perturbations to break those up and go back and forth between frog, mouse, and human. So with this, I guess I give you a reason not to talk to me or to come find me and discuss more of how you can help and get involved with some of these projects. Thank you.
So you're pretty familiar with like the methylation clocks of Synicus?
I developed those.
Do you know if it's a linear relationship? So Synicus has like a regenerative stage, and then at a certain point, it stops being regenerative.
It goes through metamorphosis. So first 6 months of its life, it's a fish, and then it loses tail and
Do you know if the rate of methylation or the pattern of methylation is linear throughout, or how it changes over that time?
So when we haven't even published this, we just deposited to bioarchive methylation clocks, which we developed for using the fact that we have different age frogs. And we did not go looking into early stages, pre-metamorphosis stages, mostly in the paper which actually establishes methylation clock. We have done different work where we're applying human developed methylation clock to embryos, but that of course doesn't answer your question. So I don't think it's known at this point.
Sure. If you had to speculate that one, what do you think you would see?
I think it would be pretty chaotic at early embryonic stages because raising and rewriting methylome is very critical. In general, we could discuss how much methylation clock reflects anything other than change in the composition of the tissue with respect to cell types. So I think generally methylation clock was meant for interpolation with the ages that you train your model for, and taking it to extrapolate is a very dangerous business.
Do you have any other questions? Or let's see here, where are we at? 5 minutes left. How's about this? Is there, for your work, is there any particular challenge or like something that somebody else could get done that would help you with your work very greatly? So for instance, for cryonics, there's like, if somebody got a rewarming process, some kind of physical rewarming process to function, that would help them. Is there something that some just a random breakthrough that might occur that would help what you're trying to do?
Well, there's sort of several chapters, right? There were 3 chapters, and for each the answer is yes. I think that largely kind of social publishing collaborative mechanism is missing for curating knowledge, agglomerating knowledge, having this sort of constructive criticism going. So some sort of platform, Wikipedia-like platform for that would be very helpful. Taking the generally distributed science to biology would be super helpful. So Daphnia platform is very nice, it's published, it is used, but there are many unsolved issues. And yes, for machine learning and for people who would program, but also for zoology, aquaculture, biology, cryopreservation of Daphnia. But the answer to your question is none of those many things I listed, but a mechanism for this sort of thing to come through. As for looking at aggregates in oocytes, I was pretty surprised that it's so difficult to measure protein aggregates and track protein aggregates. There are some imaging stains and trackers, but methods developing in this area would be very helpful.
Nice. All right, well, we're back on schedule now, I guess, and so thanks for your time, and